Okay, so welcome everyone. As I said, my name is Morgan Langille. And so bit of the format for what I'm going to talk about in the first hour here is we're going to do some general introductions. So be prepared. I'm going to ask you to just introduce yourself in a quick 30 seconds so you can start thinking about that. I'm going to give you an overview of the workshop for BMB, some tips about maximizing your experience here. And then we'll talk briefly about sequencing and some definitions just to make sure we're all on the same page. And then we'll do just some basic bioinformatics files. So things like FASTA, FASTQ files, which is usually the starting point of basically any genomics pipeline. Talk a little bit about quality filtering, and we'll talk a little bit about package managers, things like Conda, and why you would want to use them and how that helps with data reproducibility. Just some interesting fun facts here. So I did my PhD at Simon Fraser University. Steve Allen was actually my external examiner, and David Ng actually taught me a little sequencing workshop in this building about 20 years ago, where he actually had to run on the glass slides. And you've always stuck in my head as a wonderful instructor, to be honest, including like tips of how to get over a hangover. I still remember it every time. Amazing. Yeah. So very good course. And so, yeah. And actually my very first time TAing as a PhD student in CBW was here about 20 years ago in the Microbiome building just next door. And this is, I think, 10th or 11th, I always forget, of the Microbiome Workshop. So I've been doing this for about 10 or 11 years. We started in Halifax. We had it once in Vancouver 4 or 5 years ago. I don't remember, it all blurs together. We were in St. John's, Newfoundland last year, so we went coast to coast. And I keep coming back to it. And mostly because of the people. It's really enjoyable to hear about the different projects across Canada. And obviously, helping you process your data really is a big thing for me. From my own research, I do a lot of eclectic list of things. If you want to look at my publication record, if you've talked about microbiome, I feel like we've probably dabbled in it at some point. But right now, we're doing a lot of cancer work, looking at oral microbiome and blood and tumor samples. That's a whole story and a whole saga. Robin works on saliva samples from a large Canadian pediatric cohorts, population cohorts. And so she has about 4,000 samples, I think, from kids. 5,000 samples from kids and looking at mental health associations. And we've dabbled quite a bit in frailty and aging with saliva and stool. I've worked a lot on IBD in the past and probably getting back into that in the near future. So those are some of the more projects, obviously more human focused, but I have done a lot of soil, blueberries, things like that in the past. And then we also do some tool development as well. So Microbiome Helper is something that we put a lot of our lab tutorials and SOPs into and sort of constantly updates. PyCrust is, I guess, my claim to fame, was a tool that I developed as a PhD student, and you'll learn about that at the end of this workshop tomorrow afternoon, for functional prediction. And Jarvis is for connecting functions to taxa, and the same with POMS is a more statistical analysis. And Coverage Checker is being developed by Robin right now to sort of help validate metagenomic taxonomic annotation and sort of remove false positives. This is a beginner workshop, so if all of that doesn't make a lot of sense, that's okay, because we're going to guide you through some of those tools, but not all. And then the last thing about me personally, I do run the IMR out of my lab, so it's a sequencing and bioinformatics core, essentially. We do a lot of sampling, a lot of sequencing. We, on average, sequence about 4,000 samples a month right now, sort of coming and going. From samples around the world across lots of different habitats and environments. And so, seeing all that data produced all the time, makes me want to make sure that we streamline bioinformatics so that we can process data, because I know lots of people are sitting, on bioinformatic data. Yes.
Don't wander too far away from the computer.
Sure.
Side.
Yeah. On the mic.
Yeah, exactly.
Can I turn it like this?
Go ahead.
Okay, how about this? Does this work better? I feel like if I'm here, there's like this giant robot thing in my way. Okay, I'll try though. Yeah, sounds good. I can do that. Okay. So the overview for the BMB, the beginner workshop, is split up into 4 modules. So basically sort of morning and afternoon. Today we're going to talk about sequencing files, quality filtering, installing programs. Module 2 is this afternoon, and that's kind of like where you're going to probably feel a bit more satisfied and challenged, where basically you'll be taking FASTQ files and making all the way to a taxonomic table, essentially at the end of that. Module 3 is tomorrow, and tomorrow you're going to learn about microbial ecology metrics. So, like, what the difference between alpha and beta diversity, what's a peak OA and how to interpret that. And module 4, we have a bit of a few different things. It's almost like choose your adventure a little bit in there, where we'll talk about functional predictions. So that's sort of running something like pie crust on your taxa. Differential abundance testing, so finding what taxa are significantly different between the groups of your interest, and also co-occurrence networks. You're going to be getting me this morning, Robin for the afternoon lab. Tomorrow morning, Juan's going to take over for the lecture and lab, and lecture 4, I'll introduce Ryan, and Ryan's going to do a little bit of lecture, I'll do a little bit of lecture, and then we split the lab as well. So you don't have to listen to one of us drone on for too long at a time. Tap it here. It's going. Okay. So I have been doing this for a while, and so just want to give you some tips to really maximize your time here. There's obviously a lot of content on the web, right? I mean, you could, to be honest, you can even watch our recordings from previous years, but you chose not to do that. You chose to come here. It's good to all be in one place. And the big benefit of that is to ask questions, right? So I'm talking right now. Interrupt me anytime. A lot of that back and forth is really helpful. There'll be things that we bring up, and if no one asks questions about it, then now is the time. If you don't want to ask in this environment, the TAs are going to be coming around. I'll be around at all times. You can ask on a one-on-one basis as well. So there's chances to ask questions at that time, break time, lunchtime. Expand out, ask your neighbors questions. You'll really get more out of the experience. Bioinformatics, it will be a little tiring. So I encourage you to take those mental breaks, okay? So when we have a break, sometimes you'd be tempted to, I just want to crank this out. Sure, go for that if you want, but really stand up, get some fresh air, have a coffee, use the washroom. It's a marathon, not a sprint. Obviously, we're doing the basic beginner bioinformatics. If this is your first foray into any bioinformatics, you're not, I would love to say that in 2 days you'll be awesome. You won't be awesome, but you will have the skill set to, take this further. It does take time, just like any other skill set, but it's going to give you the confidence to sort of learn on your own a lot more, I think, moving forward. And then a lot of times people ask us what our opinions or, what we think is the best tool or how we should approach something. And you're going to hear a lot of people saying, well, it depends a lot of the time is the classic thing for me. So, some people view certain things as facts. You should always do it this way. And other people feel like they're opinions. But the reality is everyone comes at this field from a different perspective. And so I'm cognizant of it as well. Feel free to challenge me about things I may say as fact, and it's probably more of an opinion, but it's a bit of a balancing act there. And one of the big things I like to push a lot is, that bioinformatics is part of the research. I think we think a lot of bioinformatics as a black box that we just have to do, kind of like maybe the sequencing, right? We can just send that off, get it done, so then I can do the research. But I think you'll appreciate that bioinformatics is part of the research, right? Increasingly more so, right? Experiment design is important, but if we think about a study from beginning to end, right? Sure, design and experiment and figuring out how to get your samples, absolutely crucial. We're going to focus a bit more on the sequencing, the bioinformatics, and statistics and visualization a bit. That's where a lot of that research happens, right? So it's not going to always be straightforward point A to point B. It's going to take a lot of trial and error. You're going to do things different ways. You're going to go back. The number of times that you'll probably end up starting over with your data set and process it differently, it will happen. It's just like any project. at the end, going from point A to point B usually doesn't take that long once you've figured out how to do it. But just think of the bioinformatics as part of the research, and I think you'll appreciate it more as opposed to something you just have to get through. Okay, so that's great. So let's get into just some definitions to make sure we're all on the same page a little bit. So this is one of those facts versus opinions a bit, but I guess I'm a bit opinionated on this one, especially on certain ones. So microbiome, I usually refer to as the microbial taxa and their functions within a particular environment, right? So I sort of think of it as a sample maybe that you could sample within. If you did metabolomics or transcriptomics or genomics on it, that's all investigating the microbiome. So it's that community of microbes and their functions. We're going to talk quite a bit about bacteria, especially probably today, but Robin will get into, how we would profile archaea or microbial eukaryotes a bit. It honestly also includes viruses. We don't do a great job of covering viruses at all, unfortunately, within definitely the beginner workshop. Might cover a little bit in the advanced workshop, but just keep in mind, obviously, it's all microbes, not just bacteria. The other term people tend to use is microbiota. In my head, I just sort of think of microbiota as sort of just the taxa itself. I sort of think it as like almost like bacteria. I don't know if that's particularly the best way to do it, but that's the way I sort of use microbiota as the microbes as their taxa without sort of the functions. This one I will be a bit of a stickler for. Amplicon sequencing, Robin will talk a lot about this later today, and that's going to be the bulk of what we're processing, but that involves basically amplifying a piece of DNA. And so that piece of DNA is sequenced, and that gives us basically the barcodes or sequencing information to assign taxa to those. And we can do lots of types of amplicon sequencing, including things like 16S, 18S, and ITS, and Robin will talk about all of those. And metagenomics is more where for those staying for the advanced workshop, is where we talk about sequencing all of the DNA in a sample. And that gives us a lot more information, a lot more variety of things we can do with the data. It gives us, in theory, better taxonomic resolution. It also gives us information directly about the genes that are encoded in that sample. Sometimes it's called shotgun metagenomics. Sometimes it's called MGS. Sometimes it's called I don't know. I've seen it called a few things. My only thing here is don't call 16S metagenomics. It's a big pet peeve for me. I don't like that at all. So 16S is amplicon sequencing or just 16S. It's not metagenomics. But anyway, if you're curious about, difference in those technologies and what they cost, these are just to give you some context, I guess, of from the IMR, just like about sort of the relevant sort of sequencing approaches and their costs. So if we are doing some sort of amplicon sequencing, you're in that sort of range of maybe $30 to $35. So this is showing MiSeq or Illumina technology, producing about 50,000 reads per sample. If you want to use PacBio long reads, I'll talk about that in just a second. It's just a little bit more, although these are coming very close in price now. And then metagenomics obviously does provide more information, but it does cost more because you're sequencing a lot more, right? So for us, that's around $180. It could be cheaper somewhere else. So I'm not promoting myself or anything. And then depending on PacBio and how much you want to do for, in-depth sequencing or trying to assemble genomes from that metagenomic data, then that can scale up accordingly as well. We're not going to talk a lot about sequencing. This is a bioinformatics workshop, but obviously for this workshop, we're starting with genomic data. And so I just wanted to run sort of the two major players, or possibly three. I guess I didn't talk about Nanopore, but this is Illumina MiSeq. The one on the left is the one that's been around for a long time. And it just got sort of replaced recently with the MiSeq i100. This just came out. We actually got first one in Canada. It just arrived in January of this year. Both machines produce sort of 2 by 300 base pair length reads. And right now they produce about 25 million reads per run. And essentially what we do in a sequencing run is we barcode your samples, right? We label them with a particular DNA barcode. And then we combine those together so that we can spread that sequencing run over many samples, right? So what we typically do is we'll actually combine 496 well plates, so 4 by 96 samples. We keep one as an in control, and that gets multiplexed. We're running 360 samples on a single sequencing run, and on average, that would give you about 50,000 reads per sample. The I100 is going to double that output really soon. And so you'll basically be able to do the same. You would get about 100,000 reads per sample based on the same sort of multiplexing. And the other big thing is that it runs much quicker. So this would take about 56 hours for a single run, and this takes about 15.5 hours, the new I100. And the last point I don't have on the slide is the quality's improved a lot, especially towards the end of the read. So I'll show you some quality filtering today that's mostly from the old MiSeq, but the new ones have much higher quality right to the end of the 300 base pairs. So that's been a big staple for a long time, and full length sequencing or long read sequencing has entered into that game a little bit. So we have this PacBio Vega. We used to have it called a Sequel 2. This also just came out this year, and we also got the first one in Canada. It's been very exciting toys we just recently added to the lab. They look really cool now. I mean, look at this thing and then look at this thing, right? I mean, these are really kind of cool sequencers. They've really upped their cosmetics side of the technology. Long reads have been notorious for sort of lower quality overall. And PacBio has basically improved that accuracy by doing what they call circular consensus sequencing, or now they just refer to them as hi-fi reads. And essentially all it is that they take their long read, and this could be shotgun data, but if we're doing 16S, it could be the full length 16S sequence. So that's about 1,500 base pairs, as you'll hear about more later. They add these little bells, these dumbbells at the end, and then they just sequence over and over and over this same fragment of DNA. And basically, when it comes off the machine, they do that error correction on that single molecule. So now you basically use that long read length to sequence the same shorter piece over and over, and that improves your accuracy tremendously. And so now you're getting the same, or if not better accuracy as Lumina, whereas in the past you would. So accuracy-wise, you're getting about the same. I do not mention nanopore here. People have used nanopore for amplicon sequencing, definitely use it a lot more in genomics and metagenomics applications. We just don't run it in the lab right now, but it is another possibility from a long read perspective. Nice thing there is the infrastructure cost is a lot less compared to the PacBio. Okay, any questions about sequencing? Yeah.
Like not the same quality. Can you adjust this during your bioinformatic analysis, or no, you lost the data?
Yeah, so actually we're going to talk in a few slides about how to filter the data, but essentially we can improve it by getting rid of the poor reads, right? But we can't, up the quality after it's come off the sequencer, right? So we improve it by essentially removing the poor reads. Yeah. But you can imagine if you had all of your sequencing run was very poor, then you would either have to filter most of it or you're left with poor quality. And by poor quality, we'll see is like base by base, the sequencer will report what it thinks the quality is at that base as it comes off. It's very important for certain applications, that quality. So if we're talking about amplicon sequencing, a single base, right, could suggest that maybe it's a different strain or not. So that accuracy is really important. If you're doing more metagenomics, that accuracy can be a bit lower because maybe you're not doing that. Depends what you're doing. Sometimes you are doing strain level differences. We'll talk about that. But if you're doing assembly, then that can get sort of corrected during that process. Yep. Any other questions about sequencing? Yeah, sorry, I didn't put it on there. The PacBio runs in 24 hours now for the Vega. It's kind of neat in that you can load the next run while it's halfway through its current run. So you just keep like pushing in, and then the old cartridges shoot out the side. So you can you can run it 24/7. And as I mentioned, the I100 is down to like 15, 16 hours as opposed to it was like 56 hours before. So usually with most of these technologies, like, it produces a lot, right? Like I said, we process 4,000 samples a month and we could do more. And so it's usually the cost of running all those reagents is more limiting than the sequencer itself nowadays. Yeah. Yeah, Stephen. Yeah.
Where to begin? So one question I had relates to the input DNA itself.
Yeah, sure. Yeah.
And how in your experience that affects the overall quality of the downstream product. And potential limitations with respect to using different types of primers that clearly with the full sequencing we use different set of primers to what people are using for the Illumina platform and how that affects both quantitatively and qualitatively.
Yeah. So the primers I can punt to Robin's talk. So she'll talk about primers quite a bit. And they're in her next lecture. So talking about different types of primers, even for the same gene and how there's biases in those. So I'll punt that to her. Yeah, so there's quite a bit into going into sequencing that I'm skipping over here very lightly as we dive into bioinformatics. I guess I'll show this slide right next. And then if I don't cover one of your questions, I'll come back to it. One important aspect of this is that usually for microbiome, we're trying to get to the stage where we can say, here's the different types of microbes in our sample, right? And then we say, oh, how different are those, communities of microbes across different samples and doing visualization and statistics on that. The important thing to keep in mind is that even though we'll get to the data and the bioinformatics, there's a lot of inherent biases in getting to those lists of taxa, and it's not solved. And it means that, you could have sampled the same area as someone else, right side by side, and then you went off and did your own thing. And there's lots of inherently places that'll change your results at the end of your pipeline, right? Some of those are biological right there, like how you sample it, the tube you put it into, how you preserve it, freeze it, or if you put it in a buffer, how you prepare the library, the PCR primers you use, and then the sequencing itself. So I point to this review paper that we talked about these different factors. This sometimes is its own lecture, but we didn't have it this year, so I'm happy to answer more questions about it. Big ones are your extraction process has a huge effect on biases in your data. Freezing is overall pretty good. Yeah, like -80 storage is pretty good. But if you compare that to some sort of buffer solution, there will be differences between those. The sequencers themselves technically are quite good. So we don't even worry about run-to-run biases like we used to. It's pretty consistent for what we're looking at. And then obviously more so in this workshop is, thinking about the bioinformatics. But in theory, the bioinformatics you can control, right? I mean, you're going to run hopefully the same pipeline. But just keep in mind that if someone else, you read a paper and they had a similar sample, their methods are going to obviously change, what they're going to report. And so how they assign taxonomy, how they do their filtering, all those things will lead to shifts in what they're reporting to you. If you have the data yourself, then obviously you can put it all through the same pipeline to avoid those differences, but it is quite important. So did I answer all your questions?
I just wanted to get it out there. Yeah, that's good.
Yeah, okay,
I mean,
okay.
You had addressed that in the review, but it's just that I've seen how batch effects can accumulate when
Yes.
you're getting samples from different sources of different DNA qualities and yields. And then, you don't realize until you're trying to put all the data together that you may have to throw samples out.
Absolutely.
Downstream.
Yep. And then I think I mentioned it later, but we talked about, keeping track of some of that information to try to account for it, at least in the bioinformatic, and look for those biases is important. And then the quality of the DNA is really important as well. Overall, like Illumina is quite resistant to the quality of the DNA, meaning it can be sometimes quite degraded. PacBio or other long reads have been notoriously a bit more sensitive to that degradation just because you need a long read. So just from a pure like fragment, sometimes they get fragmented up. And also in the past, usually Illumina requires very little DNA in their library preps, whereas again, PacBio and Nanopore often require more amounts and more cleaner DNA, but the library preps continue to get a lot better on that side as well, much more robust. Yeah. Yeah, I think the classic saying crap in, crap out applies to sequencers as well as bioinformatic applications. Yeah. Okay, so that's any other questions about the sequencing stuff? I'm happy to ask questions or answer questions along the way as well.
What's your facility called?
Yeah.
The price list you put up there?
Yeah, it's the IMR, it's the Integrated Microbiome Resource. Yeah. Yeah. It's a it's not a company. I know it feels like I'm pushing it a little bit. It's just like a sequencing core in my lab right now anyway. Yeah. Okay, so now we're gonna really back off all that sequencing and stuff and get really bare bones here. And so we're gonna like start at like 101 bioinformatics here about Unix and types of files a little bit just to make sure we're all on the same starting page. So this is a bit of a dry slide, but it's important. So when we think about files in a computer, right? A lot of times we interact with files in Windows or on a Mac or whatever we do in a very like, it's got a nice little picture of it and we just put it into a program and it works, right? When we're doing bioinformatics, it's much more bare bones. Like obviously we'll be typing into a command console window and we're interacting with files in a very rudimentary way. In most applications, the files we're interacting with are just plain text files, meaning that we can just open them in anything that can read a text file, right? And that's nice in that sometimes it's just nice to look at the file and see what's in it to make sure something hasn't gone wrong along the way, right? It's classic where people would have your sequencing file and you do filtering and then you move right on the next step and then all of a sudden you get an error and then you look back and you're like, oh, why did I get an error? And you look at your file and it's empty, right? Because you're filtered everything out, right? So, you'll get used to sort of opening them and looking at them. Sometimes it's hard to look at, it's a lot of data there. But often we can sort of investigate that. And then there's essentially different command line tools to analyze that data very easily, those text files. So the same files we use to store DNA are, files that run computers or anything. And so we can look just at the top of that file with something like cat or head. We can run an editor like nano. We can count the lines in a file. And so the other important part of that is that file suffixes. So when we see that .txt or .tsv or my favorite program, whatever it's called, they're just like a suggestion. Often in Macs or Windows computers, that'll prompt a particular program to open it up and take it away. Here, it just sort of gives you a hint to be like, oh, okay, this file likely has this type of data. But when you run the program on it, they don't usually ever look at the suffix file. It's almost just like an optional thing. And so it's really a hint of what should be in that file. The sort of alternative to a plain text file is like a binary file. And so that has its advantages in that it's usually takes up less space. It's usually faster to sort of parse in some ways, but it requires a specific file to open up and read that binary file. And if you attempted to open it or look at it, it's going to just have this gook of letters that will just mess up your console and it'll look really messy, right? So it just looks weird. That's fine. There's binary files that you will use in bioinformatics as well. BAM files are like an alignment file where the B stands for binary, right? And so there's advantages to those. And because we are working with fairly large file sizes, sometimes we have to move those around. And so it's just good to understand the idea of zipping or compressing these files, right? So a plain text file by itself isn't very space efficient. It's easy to look at, but it does take up the most room. You can compress that easily with various different tools out there. You've probably come across zip files. Often we'll see gzipped files in Unix world. And essentially all that does is it converts that plain text file into a binary file. And so that just means you can't look at it directly, right? Without some shortcuts. I mean, you can sort of do it without uncompressing. You'll learn those one day, but you usually have to uncompress those files before then proceeding on. But really the whole purpose of compression is just to save on storage space. Yes. Oh, it's just the type of zipping technology. Gzip is like I think it stands for maybe GNU, which is a more Unix open source zipping technology. I don't know who made the original zip. Any ideas, TAs, or anyone, the difference? No, they're just different approaches. So you couldn't take the tool to unzip a gzip file would not be the same as a zip file. Or there's other compression technologies as well out there. Yep. Yeah, the details of that, you probably don't need to know. But they definitely like a trade-off between speed and compression. Okay, and so what would one of these plain text files look like? So this is the classic sort of text file for representing sequences. And so this just gives you an idea of, you'll have files like this in your analyses. And so this is called a FASTA file. It comes from Fast Alignment, which was this old school alignment tool from a long time ago. But the important parts is that it's a plain text file. And the only part of its formatting essentially is that it has a header, which is identified by this greater than sign. And then anything after that greater than sign is just a identifier. So I have my gene here. It could actually have a gene name. It could be a very long identifier. Sometimes those headers are very long. Sometimes people will put tremendous amounts of data in those header files, like it's crazy. But it just has to be some sort of identifier. In ours, it's usually just a sequence identifier coming off of the sequencer. And FASTA files can be both DNA, which is shown in the top one here, or a protein sequence down here. And so this is just showing the DNA sequence, and that is just a series of obviously nucleotides spread out across multiple lines or, a single line. Yeah.
This one that you use, it shows sequencer sample, is this file that you obtain then, right?
So the sequencers will give what I'll show in the next slide, which are FASTQ files. And FASTQ files have quality information and are a bit more tied to the technology. Often you will take sequence files off your sequencer, and at some point you'll likely have a FASTA file because it's just the most simple, universal way to sort of pass sequence files around.
All of them generate the same kind of format, like FASTQ, you say, right?
Yep.
Can I compare different forms together, even though the primary sequencing or like if they are different?
Yes.
Can I compare different forms? For example, I'm doing one project in Brazil and you in Canada. So I use one platform and you use a completely different one to do your sequencing. But if we have the same FASTQ file, can we compare? They are comparable or no, I cannot compare if they come from different technology.
So you could technically run the same bioinformatic programs on the data, right? So often what will happen is you'll often do sort of unique filtering to the sequencer, maybe. You'll get it to a FASTQ or FASTA file, and then most tools will then accept those files throughout, and you could technically run the same commands on all of them. Now, should you actually compare those to each other, it comes back a little bit to those biases, right? So sure, you can run the same bioinformatic processes, but you have to keep in mind that the primer choices that you used, how it was extracted, how it was collected, would possibly alter, obviously, what you're getting out of the sequencer. Yeah.
And noise can be eliminated by filters or no?
Oh, the ability to correct for batch effects it depends on the type, but it is difficult. Yeah. And it sort of depends on how you want to compare them. but it's difficult. Yeah. For example, an extraction. So you run a different type of DNA extraction process. You do QIAGEN and this person does some homebrew method, right? That inherently is going to amplify or extract. It's going to extract, different microbes better than others, right? So you get more DNA from some microbes than others. And that will come out through your sequencing so that all of a sudden now maybe you're missing a particular bacteria because the extraction process wasn't robust enough to break down that cell. That's an example, right? And so there's no bioinformatics that can kind of overcome that. Yeah, there's no like, hey, did you use QIAGEN and use this? So now we have this bioinformatic tool to correct for it. No, that doesn't exist. I mean, people have attempted, don't get me wrong, but it's fraught with possible issues. Yep. Yep.
Do there have been studies that compare results across centers using the same source materials?
Yes.
And I remember years ago seeing that there was a batch effect by center.
Yes.
Has that gotten any better?
So I don't think anyone's done it recently. And I think it depends what you're doing. So for example, one of the arguments about sometimes metagenomics, and actually clinically why it's relevant here a little bit sometimes, is that for metagenomics, say with Illumina, most, not all, use the same Illumina library prep, Nextera, Flex, or whatever it's called nowadays. That then removes at least that sequencing center bias possibly. With 16S or amplicon sequencing, you'd have to use the same primers. But again, depending on how they exactly order their primers and what oligos they buy could differentiate across sequencing centers. So again, you might be doing the same variable region, but you would get a bias in that sequencing because maybe they didn't buy the exact same primers. So yes, I don't think there's an inherent, my feelings are there's not as much inherent bias in the actual sequencer itself. I could be wrong there, but I feel better about that. Yeah. Okay, good, great. Okay, so FASTA files, just coming back to the basic bioinformatics, you'll see these with like various sort of applications. So we'll be dealing it from a, hey, we're getting a read off a sequencer, and each one of these entries in a FASTA file, like you would have multiple, 1, 2, 3, or whatever your identifier is, would represent a single read off that sequencer. But like other applications will actually use FASTA files for other aspects, and they'll actually add these suffixes. So an FNA file means it's usually a nucleotide format. Often you'll see that used for like a whole genome. And so this, I say gene here, but it could also be like the whole genome of an organism as a single entry. So if you assembled that whole genome into a nice one big circular piece of DNA, that could be represented as a single entry in a FASTA file. So you'll come across these types of files if you go to like any sort of sequencing database like GenBank and then CBI, you'll see these files there, and the format's the same, but sort of what that data represents within it will vary quite a bit. Okay, wonderful. And as alluded to, there's FASTQ files. So these are the ones that are sort of like FASTA, but they include essentially quality information coming off of the sequencer. And FASTQ files were quite standardized for a while, and I find they've backed off of that a little bit. This is showing one example. It's not like a FASTA file in that there's no greater than sign and there's no header and information. Instead, it splits basically the information over 4 distinct lines, okay? The first line is some sort of identifier information line. And this is quite unique to different sequencing strategies and sort of how to parse this. The second line is the actual DNA that's been read by the sequencer. This third line sometimes uses this plus or minus, representing in theory a strand. It's not very much used at all anymore in FASTQ files that you'll come across and is usually not meaningful. And then the fourth line is the actual quality score for that base pair above it. This encoding in the bottom changes over time depending on FASTQ formats and on the sequencing strategy. So this quality scores, and I don't go into it, but there's different types out there. You can basically find out what that represents, and then programs will interpret that, which you then use to filter the data. Okay, so for example, I actually don't know here, but imagine E's are like sort of represent a FAST quality score of 30, and then this greater than sign means something less. It doesn't really matter in that you'll never try to interpret this line by looking at it. It just means that the FASTQ file has that information, and programs that you'll run on it will take that into account to figure out the quality at each base along that read. So it does mean that if you have a FASTQ file, you can convert that to a FASTA file, right? But you lose that quality information and you can't go back. You can't go from a FASTA file to a FASTQ file. Great.
E is 36.
What's that?
E is 36.
E is 36. E is 36. Well done. That's good. Yeah, I don't know where I got this from. For reference, 40 is like the max score. Yeah, so 36 out of 40. Why? I don't know.
Is the max score information. I think some of the sequencing video sequences go up to like 19.
Really?
Yeah. They're violence.
Yeah, I don't go into it too much. I'm trying to avoid not too much sequencing stuff, but it's also like Illumina recently with the I-100, I know has gone to slightly more staggered qualities. So instead they used to have basically every quality from like 0 to 40. And now they have essentially like tiers within that. So essentially they've grouped, I don't remember the grouping right offhand, sorry, but like 35 to 40 is now one quality score. And then they have different ranks. I'm mostly mentioning that it does change over time and you have to be cognizant of whatever quality you're parsing. Okay, so this is a very this is so bioinformatics right here, right? So this is a file name and a path. So you're gonna start to like process files. Some of you hopefully have all done maybe the little Unix primer maybe, or if not, you're gonna learn that in the next module quite a bit. But when we look at a file like this, there's some things that should be able to almost interpret a little bit, right? So really basic Unix means this squiggly line. Anyone remember what a squiggly line means with a slash like this?
Oh,
Home directory, that's right. So that means that we're starting in our home directory, right? And that this data represents a directory, right? So now we have our home directory, data. You can imagine a little folder window or whatever you want to imagine. And then within that, there's another directory or folder called intensities, then another one called base calls. And now we have this big long file name, right? And often you'll see sort of this encoding. We have maybe a sample name. It could be meaningful to you. It doesn't have to be. It could just, it could represent maybe the file name that you wrote on your little tube, right? Your little sample S1, I don't know, whatever you use. And then this can come off sequencers. Sometimes with Illumina, you see things like this that represented lanes. I'm not going to go into that. R1, I will leave forward and reverse reads to Robin, but this means the read one is the forward read on Illumina technology. And then 001 is also often just coming off Illumina. But now we have some suffixes, which are, again, hints at what's in it. So we know that this is what? A FASTQ file, right? Has that quality information. And it has a .gz on the end of that, which anyone want to guess what that means? It's gzipped, yes, so it's compressed. So we can't just, open it up or look at it. It's going to be all binary, right? So if we want to look at this file, we'd have to unzip it. We'd have to run gunzip on the file. extract that information, and then we could look at those FASTQ reads within it, or then process it. Okay, so that's great. What would we actually do with that data, right? So we've run our samples, we've gotten the sequences from the sequencer, it has that quality information. Then the first step of basically any pipeline is you're going to start thinking about filtering, right? The sequencers, depending on what you run, already do some filtering, right? They're already doing stuff in the background. They're basically throwing away some things, but you're still going to get reads. And no matter what, you're going to want to take a look at them. You're going to want to see sort of what the quality looks like. And this is just showing a report created by a tool that you'll run today called FastQC. And this just shows the quality, higher is better, and then showing on average with box plots across all the reads, the sequencing quality along the length of that read, right? So this is showing a 0 to 300 base pairs. This is on Illumina sequencing technology. As I mentioned earlier, it's very classic that you get this drop off, especially in the last 50 base pairs. This is actually considered quite good. Sometimes you would see sequencing runs which really dip down. And then that information gives you, one aspect of what the quality of your sequences look like. From a filtering perspective, you could think about trimming, meaning that you would start to remove maybe some of these base pairs at the end, or you can think about filtering in that you're removing some of the lower reads, right? Lower quality reads so that the overall average is higher. You can also compare that across your samples or across sequencing runs to compare. And this is just simplifying that now. FastQC averages across, say, multiple samples to see if there's some, batch effect or something going on with the data. You get this report along with many other graphics out of this FastQC file. It's actually one of the prettier programs that you'll get data of. Usually you run, complex bioinformatic pipelines and you just get a nice table. FastQC is nice in that it'll actually produce like a graphical output. And there's actually a lot of other graphics that will output as well about overrepresentation of particular sequences, notorious primers that are overrepresented. There's lots of other metrics, and you'll see those in the module. Okay, and how are we doing for time here? I'm just talking. Are we good? 9 to 10? Is that when I was supposed to talk? Yes. Great. I'm doing great. Well, sort of. Okay, so quality filtering, like what should you do? There's lots of options out here, and a lot of filtering is sort of built into maybe different programs that you'll run. So you could use its own independent program, or say if you're running Chime later, Chime has some built-in filtering pipelines. There's different strategies of how to filter, right? So you could think about every read has to have a minimum base quality, say on average of Q30, or maybe it's like every single base has to be over Q30. You could have a percentage of the read. Sometimes sequencers will put out ambiguous bases, like meaning it'll actually put out an N instead of an actual letter. And so people just don't want to deal with that. And they'll just literally toss the whole read, which is fine too, depending on, if it's a small fraction of your sample. Usually the thought is that filtering is a good idea, right? You might as well have less sequences of higher quality than more sequences with lower quality. But it's a trade-off between the two. Yeah. Yep.
Like there's a pattern. Each line is a sample, right?
Yep.
And the way that the samples goes up and down looks very close. That means that your noise is equally distributed, or your error is equally distributed. For example, for the filtering, or no, this is not related with the quality of
Yeah. So in those cases, they're quite tight. If you look at box plots here, then obviously there's reads outside of those averages that you would possibly be removing to increase that average and increase that quality overall.
But if you see the other one, look like they are pretty much like the same.
Well, that's the average.
Yeah.
That's the only average, yeah. So it's possibly a little misleading. Yeah. Yeah, and then so there's ways to filter for quality. If we're doing a primer-based thing, often you'll look for that primer to make sure that that reads in your sequence read itself. And it's not some spurious PCR product. And then you'll often trim that primer sequence off of the read. And there's lots of tools out there to do types of different filtering, Triomatic, Sickle, Cut Adapt, or like I said, they'll be built into the pipelines that you're sort of using. So filtering, yes, you should do it. Two, it varies quite a bit. And three, it will depend on the sequencing, unfortunately, right? So you can't just be, I'm always going to apply this exact cutoff all the time. You know, if it throws away 80% of your data, you might have to be a bit more lenient on it, right? And so that's just the reality of sort of looking at the data, looking at the filtering, and coming up with what's acceptable to you. Yeah.
Reverse reads.
Reverse reads, yes.
Yeah. How often do you find that people work with them?
Yeah, so I think Robin's gonna talk about, are you talking about paired reads, Robin?
I'm pretty sure about them.
Oh, okay, well,
I mean, yeah, I've talked about it in practice.
no, it's all good. Yeah, so for people who don't know, the Illumina technology produces a forward read and a reverse read from the same piece of DNA. Yeah, so I'm not going to get to the logistics of it. It's very cool. It pens and then bends over and it amplifies both. But the reality is you get a forward read and a reverse read, R1 or R2. And this is specifically for Illumina, okay? And so those, depending on what the fragment of DNA is, if it's a large fragment of DNA, those reads may not overlap, right? But you do know that that piece of DNA in between is connected. So that's useful for actual some algorithms down the road. You learn about assembly later. That will come into play. It's very important. If we're talking about amplicon sequencing, we often develop the primers so that those forward and reverse reads overlap. Yeah. And because they overlap, you know, in theory that that piece in the middle where they overlap should be the same in theory, right? And so you can, I think, Ron, we'll talk about stitching, we call it, where essentially it stitches the forward and the reverse read together to make your one read. That fragment of DNA, and depending on the overlap, in some cases, in many cases, it'll improve the quality overall because you have the forward and reverse read to somehow get some agreement between those, and you'll use those fragments. Others will sometimes not even use the reverse read because the reverse read in the past has often been lower quality overall. And so sometimes people will say, look, my reverse read is just awful looking. So I might just discard the reverse reads and only use my forward reads here on out. Again, it kind of comes down to your PCR product, the results of your sequencing, and how you want to move forward. Some people are very, we have a client that's very like, everything has to be super high quality. And they're like, just crazy. And then other people are just more relaxed. They're like, they're happy. Usually, I guess for most of our pipelines, we stitch the forward and reverse together and we use then those reads moving forward from that.
DNA very fragmented, it's better to use this technology, like for example, from paraffin samples.
So
If the DNA is very,
if you have fragmented DNA, I mean, short read technology is a bit better because it's shorter and but you are amplifying up a PCR product, right? You're not going to get a fragment of that PCR product in that case. If you're doing long read sequencing, there's no forward and reverse read, right? It's one big hi-fi read. And so you don't even have to worry about that process for those. And in theory, you get the whole gene. And nanopore, I'm not as much of an expert in that. So I think they used to be able to get like sort of a forward and I don't remember. It's mostly one, right?
Yeah.
They used to have a 2D sort of thing at one point.
Saying on school things.
Yeah.
In practice.
It's 1D. Yeah. Yeah. Thanks. Does that answer your question, Stephen?
Yeah, it's just, it comes up a lot, like when you're working with this data, especially from public resources.
Yes.
You might be shocked to learn that there's only the forward read
Right.
available. Their microbiome is notorious for that.
Yep.
Where they only publish the forward read data. But they talk about having both reads. So if you're looking to pull from the trace archives to do a comparative study, you might burn cycles looking for this reverse read that you've been asking for, but isn't available in public. Just, something to keep in mind.
Yeah. Good. You did.
I just had that general question, but obviously the higher the Q, the better.
Yeah.
But do you have any comments on what's good enough? Like, so for instance, we use MinEye because from a teaching perspective, the infrastructure costs, mistakes are lower when students are using it.
Right.
But their Q value 20.
Right.
Is what they still 99%, which I would imagine is good enough for anything, but do you have any comments?
Right. So if we're talking about amplicon sequencing, right? 16S sequencing, then you have to sort of keep in the back of your mind that then if your Q value is lower and you're expecting, say it's 99%, right? So now you have an error like 1 in 100. But the fragment that we're often amplifying is, say 400, 500 base pairs, right? So now,
15 or like the full.
oh yeah, sorry,
Yeah.
that's full length. Yeah, of course. So now you're going to have, a lot of possible errors in that read. What does that mean? It means that, you're going to sequence possibly the same organism and get different reads, as many as 10 nucleotides or more difference between those. And at that point, you would have to think about clustering the reads, right? Like Robin's going to talk about ASVs and O2s and clustering. And so you wouldn't want to interpret, a read that is 5 base pairs different than another read, 5% nucleotide density, as a different thing, right? So it means then your resolution essentially is a bit lower, right? You're not going to be able to say for sure if this is a different species or not, or possibly strain or something like that. Yeah. For metagenomic applications, maybe not as big a deal, because again, it's depending on what you're doing. Yeah. So I think it comes down to how you proceed. Yes.
But for most of it is kind of a balance between, do you care more about having highest quality read or having more information available? Because if you only have a short read, you're a lot less likely to be able to classify it well taxonomically. And I looked up what I was saying about low quality score. The newest PacBio would just go up to like 90.
Oh, thank you.
That's impressive.
Yeah, and open pipelines aren't really designed to filter it.
Yeah. Okay, great.
Okay.
Just kind of building on that statement, there's also the issue of when you're clustering, if you're using the lower Phred scores, you'll wind up, let's say you're using ASV, you'll actually wind up with a much longer tail in your distribution. And that will affect other downstream analysis that you're doing for indicators. You'll wind up seeing things that you're hallucinating. And so it's probably best to use a higher Phred score and lose a little bit of data so that you can improve the overall statistical quality of that distribution.
Yeah, and remember I said often you'll sort of not go from point A to point B, you'll go back. It's not uncommon for the people to be like, I'm going to do this filtering cutoff, do all their bioinformatics and be like, I wonder if I filtered too much. I wonder what happened if I backed off my filtering, and then like make sure that your end results are robust, right? I mean, so this is not like I'm going to do this thing once and then move on for the rest of my life. Maybe it works that way. I mean, great if you can do that, but often it's pretty common that you might have to go back and forth a few steps. Okay, so that's sequence files. And then the other major component of any sort of bioinformatics and definitely biological interpretation is the metadata file, what I call the metadata file. And this basically provides context about your sample that those reads are coming from, or about even the project itself. So if we start first at the project stage, This is really important for, most projects. You're going to upload your data into a public repository, and there's different criteria that you can add to it. I was part of this consortium that's sort of for human microbiome research, at least having a STORMS checklist of criteria to make essentially that data more useful to others when they want to collect it and do something with it. There is other checklists for projects for non-human microbiome data. And it's still pretty much the Wild West, though, of what people choose to upload with their data, unfortunately. But, you could think about, obviously broad things like, hey, is this an ocean sample or a mouse sample? That's really critical. You can imagine, though, knowing what type of extraction method went into the processing of it would be really useful. Do most people put that in there? Unfortunately not. It has other things about where the data came from. If it was actually from an environmental sample, often you will get the geographical location, which is really cool, so that then people can put all that data together and sort of like look at trends around the world, which is very fascinating stuff. So that's sort of one level of metadata at the project level. But often what we'll be thinking about is what I would think about as a per sample metadata file, right? So you have basically each sample that you're going to look at, each with its community of microbes, and then you're going to have essentially information about each of those samples. And you put that into some sort of table, simple as Excel, but you'll often convert that to a tab delimited or comma delimited file. And that's going to be important for once you process the data to interpret it properly. And so it's usually a mix of both the thing that you're really going after, right? Your experiment design, case control, if you're after, does this depth of soil change to this depth of soil, then that's the sort of grouping that's really important. But often you're collecting information as things you might want to investigate along the way, or at least take into account as a confounding variable, right? So if we're thinking about a humans study, maybe you're comparing, some sort of ulcerative colitis versus a healthy control, right? But then you'd also want to know about their age and their gender or sex, possibly. Maybe you'd want to take into account other factors about that individual. Same with mice. Maybe you'd want to know what cage they came from, right? If you're doing environmental, maybe you'd want to know who sampled it, right? That's more of a technical variation. And then, as I was alluding to before, you can keep track of the technical side too, right? When was the sample collected? Was there some sort of weird batch effect maybe on days or not? When we get into the lab, it's nice to track, extraction groupings. So if you're doing a whole plate at a time, tracking what sample was on what extraction plate to make sure that that didn't possibly confound it, because you can look at the data after. If you're doing mice, that'd be sort of in that bit of a technical thing. We're looking at cage effects. Usually more data is better because you can always just choose later what you're going to look at, right? But once you've got the samples, you can't often go back and figure it out, right? So, yeah.
Where do you keep this metadata, and what would you like factor in, like the FAIR principles?
What are FAIR principles?
Like the findability, accessibility, interoperability, and reuse of digital access. It's like making this information like accessible to others. It's
To others?
yeah, that's like a requirement for like granting and things like that. That you have like a data management policy, things like that.
Oh, I see what you mean. Yes. I haven't come across FAIR before. I guess I usually take it from account of grouping these into two things, like one about the project itself for context, and two, usually what happens for our projects is we try to keep track of as many things as possible. If you're doing environmental or non-humans where the ethics is not a concern, then you can basically keep that table and add it to lots of different locations. Obviously, it can be linked to the publication. You can have it on a GitHub site. And depending on the repository, you can encode some of that data into those public repositories as well. If you're doing humans, often, unfortunately, what happens is there's a lot of ethics, and so they limit, sort of due to privacy, what you can upload. It's just very, very frustrating. And so you sort of have to, you're usually, you would write that into your ethics from the start, right? About what you're allowed to publish. Yep. Yep.
We are at.
Okay, we are at 10:30. So let me just walk through a little bit more. Is that all right? Yes. Everyone good?
Keep going.
Great. Okay, so here's an example of a metadata file. This is actually like sort of what you would get from a, hey, I'll type less or more into my Unix terminal and then like look at the output. But you could also obviously have this loaded up in your favorite spreadsheet program. Usually, not always, but the first one of the columns is going to be your sample IDs, right? It's going to link your actual file name that you had to this file with your metadata. And then you just basically add columns for the different types of metadata that is relevant to you, right? And that encoding within each of these columns is really up to the type of data. So this is an example with different samples from a mouse study. Source indicated 2 different institutions where the mouse came from. So these are meaningful to me. This is New Zealand and this is Halifax. These are cage numbers that the mice were in. And then genotype is the actual thing we were testing. This is a wild type mouse versus a particular Chimera knockout mouse. And so those things can be groups. You could have 2 or 3 more groups. It could be a continuous variable. It could be like water depth or pH or whatever you're interested in, right? But essentially, most bioinformatics programs are going to take that metadata file and you'll often operate on, hey, I have my samples, group them according to this metadata column or stratify it and then run an analysis to either color it or do statistics on it or look for correlations. Okay, starting points, really important. You'll see that some will combine these files like into a biome file, so it's kind of convenient, but the reality is they're quite distinct things, right? Okay, so that's that. Any questions about sequencing or metadata files? This is great because we got a lot of sequencing in there, which we didn't cover anywhere else, so that was good. Okay, then I think I'll go through this pretty quickly, this little bioinformatic packages. It's not as, I don't think, question raising, so I think it'll be pretty quick, and then we'll get a break in. So installing bioinformatic programs, just to let you know, is non-trivial. It is not that easy. And so often once you leave here, you know, banging your head against the wall trying to install a bioinformatic program for a day is very normal. So don't feel like it's you, it's the program itself. As part of this workshop, because that is a time-consuming process and we can't have all of you, unfortunately, do all of those installations of all those programs, we do a lot of it ahead of time for you and save it in an image. So it sort of helps speed up the process. But it's important that, you know, it is challenging and it does take time. Installing programs can be sort of just Wild West of like, hey, I'm just going to find this command and then install it quickly. But there's more organized and robust ways to install programs and their dependencies. And so really this comes from the fact that we all build different tools, we, as in people, bioinformatics or whoever, and they build on dependencies from others. So if you want to install software A and run it, well, maybe that depends on software B and C, but B depends on D and E and so on. And so those are dependencies. And so if you want to run a particular software, you need all the other programs installed with it. Of course, some people will package that all together nicely for you, but it's not always that simple. And then the other side of this is that if you change versions, you can imagine now, oh, there's a new version, but it uses different dependencies. Well, now I have 2 other versions of the dependencies, and that is quite confusing as well. And so the important thing here is that there's different package management systems, essentially. And so this is a large area, but I just want to introduce this idea of like why that's important. We use Conda a lot in this workshop. And Conda is a type of management system to essentially package those software technologies together and allows you to have multiple environments so that you can operate different versions. I think that's basically what this is. And so with Conda, you can basically make this virtual environment. And then within that virtual environment, you have different packages. So you could have different versions of Chime, which I'm showing here. You could have different softwares within each of those versions, and you're able to make new versions all the time. There's different types of Conda. I don't think this is actually too super relevant right now, so I'll just blaze through it. Anaconda includes Conda plus a whole bunch of R and Python packages. It's very large, very large. And so people develop mini Conda, which is quicker to get off the ground and, only includes Conda and lets you install programs. This is showing a few, just creating a new environment called My Environment. And then you would install packages within it. And it just means then when you, first log into your terminal, you have to activate that environment, right? Or if you ever want to switch environments, you can also switch between them. Okay, so say if you type a command that says command not found, it's probably because you mistyped it or you didn't activate your environment to start. And then just as a fun other thing is Conda is sometimes slow at installing packages. It can take a while to resolve them. And so Mamba in theory helps speed up that process. It is a direct replacement for Conda, so it's nothing really different. It's just in theory quicker and faster. And then YAML files. So in many times when we install packages, they're in a public repository and Conda knows where to look for these packages. But there is ways that essentially you can get a bioinformatic package that releases its software as a YAML file. And that's basically like a recipe or instruction book of what softwares to grab to make your software run. And so, for example, Chime, you can just literally grab this file. So wget means that you're going to download this YAML file. It's actually a plain text file, just coded interestingly. And then this shows how we would create an environment with that file. And then this would grab all the dependencies. We don't need that afterwards. And now we can activate Chime and you're away to the races. So overall, package managers are good. They help you organize things from a. Yeah, I find a lot. Sometimes you'll be running a project. You'll start a new project, and maybe you want to have a newer version of the software. You can imagine keeping the old software around as well, right? So that you can have that data reproducibility. And there's other package managers out there, and then there's also other types of things like this. So if you've ever heard of containers, containers are kind of like Conda, but like more elite, no, more beyond it a little bit. And what they do is they basically allow easier sharing of the programs across platforms easier, and then you can run programs almost directly with the Docker image. With the container. There's different types of containers. Docker is probably one of the most well-known ones. Singularity, I guess. The people in the back know this technology way better than me. I'm not as comfortable with containers as I am with just Conda. But I think you'll probably come across it. And essentially, though, the idea thing, keep in mind is that it's a way to install a program, in theory, a nice, easy way that keeps it encapsulated. Yeah, and it's not going to mess up others. Is that fair in the back? I know this is a super high level. Okay.
It's app container, so it's Docker and
App
container.
Apptainer. Thank you. No more singularity. Okay. That was confusing anyway. It's like, anyway. Okay, and this is my last two slides. So the other side of that as well, beyond containers and sort of helping reproducibility, is tracking what commands we run. Yes, so if you've ever been in a wet lab, you might have had a wet lab documentation book, or maybe you use a file thing on your computer or something. Bioinformatics is the same way. It's very easy to blaze forward and just mash away at data, but trust me, you're going to go back and you're like, oh, I don't know. Or your supervisor or someone else is going to ask you, what filtering did you run? And then you're like, I have no idea what I ran because I didn't write down the command and I can't even go back to look at it, right? If you keep track of what you've run, you can easily go back and change things and tweak it. And plus, you'll you won't have to relearn it every time, right? I mean, you can mash the buttons and it'll eventually maybe run, but then you're not going to like do that over and over again. So reproducibility is important for bioinformatics, and it's going to be good just for you all around if you take that extra time to write them down. Now, beyond just writing them down, because you're not going to write them down, like that's craziness, right? I mean, don't do that. There's ways to facilitate that, and there's even some really nice ways to sort of organize it. So GitHub is technically a way to version control software, but there's these things called notebooks. And so there's R notebooks and Jupyter notebooks. And so hopefully you can see this. You will be using a R notebook, right? Okay. Okay. But anyway, what this is showing is that you can divide it into sort of the left-hand side and the right-hand side. Sorry on the size here. But what you can see on the left-hand side is that you can encode basically some text, right, with certain labels like title or whatever that says there. And then you have a way to encode your actual commands that you ran, right? And then you can have some more comments and things. And the cool thing is that then the notebook translates all of that and sort of in real time and presents you with a nice formatted notebook. And it even can show you graphically what was the output of that command, right? And so that means, and you can even edit it within that nice graphical version later on. So it's a way to keep track of it, organize it, and sort of move through the data analysis in a very robust and reproducible way. Yes.
Obviously, it's important to keep track of your code for yourself, but you're often required to publish your code when you're publishing a paper. So regardless of whether you think it's important to do for yourself, if you're going to remember everything, you have to have it.
Yeah.
Not really.
Yeah, I think pretty much it's mostly accepted. Yeah. And I would recommend often, if you are going to publish it, just make a little GitHub account, put the paper in there, and then you just have the code. It doesn't have to be beautiful. No one's going to judge you. Like, no one's really going to even look at it that much. And like, if it is, it's just someone just like you. So like, don't worry that it's messy. And if it's functional, it's good. Yeah. Yes.
Yeah, just a question, what's the difference between our notebook and our markdown?
Our markdown is the actual I guess the syntax. See this? I don't think syntax is the right word I'm thinking for. It's a way to encode the text, which is then interpreted. And then Notebook basically takes that R markdown and graphically lets you sort of see it. I think it's the best way to describe it. Is that fair, I think, for everyone? Yeah. So R markdown is just the actual syntax of like, hey, bold it by typing this way. And there's lots of different types of markdown out there. And they're actually quite similar. And then the notebook just builds upon that so that you can take your markdown and interact with it through this graphic visualization. Okay, sorry that went a bit longer. Really good questions. The sequencing stuff was great, so that added to the conversation. I'm actually happy about that. But we do have a break. So,
to extend the break a little bit, maybe come back at 11:15 instead of 11.
Yeah. Everyone like that? How about 11:10?
Okay, so we're gonna say, feel free to grab coffee and food, or use the bathrooms, go on an adventure to find them. If you were not able to connect to the AWS session, we'll cover it a little bit next time, but also feel free to call me over and I'll give you some specific help.
After the break, you're gonna be doing this, so it's gonna be exciting and it's gonna be a lot more interactive here with people helping. So get that caffeine, food, whatever, get ready. All right, great. Okay, thanks.